Hello, and welcome to this week's Ethical Dilemma in Science. For this week, I want us to start out by imagining a situation where you have a mother and father that are interested in having a healthy baby, but there's a genetic condition involved. There is an inherited disease that, although it's variable by the person that has this disorder, it is usually associated with very substantial impairment and a pretty severe reduction in life expectancy. And so this, I've got a screenshot here from the Lilly Foundation, and this is a foundation that was formed by a mother in memory of her infant daughter who died of what's referred to as a mitochondrial disease at 8 months of age. So an example of this situation that I'm presenting to you. Now, you've got this situation, you've got this inherited disease, but now science has a possibility of basically having a healthy infant, but it's going to involve getting DNA from a third person. In essence, you're going to be forming a baby that would have 3 parents. So this week's Ethical Dilemma in Science is titled, Heather Has 2 Mommies and a Daddy. And we ask the question, should in vitro fertilization use DNA from 3 parents in order to create a healthy baby? Now, I titled this talk, Heather Has 2 Mommies and a Daddy, in reference to the 1989 children's book, Heather Has 2 Mommies, which shows a non-traditional family structure. Now, this book that, again, came out in 1989 is a very favored book to be on banned book lists. According to the American Library Association, it ranked 9th most banned book in the 1990s. And I'm a big proponent of obtaining information from diverse sources, and I'm concerned about the view of banning books and banning information. And so using this as a starting point, this book is highly controversial, and what we're going to be looking at is a medical treatment that could be controversial as well. So like all of these ethical dilemmas in science, it's based in part on current events, and this one is no different. What we're looking at here is a screenshot from the U.S. Sun article from May 2023. Three's a crowd. First British baby with 3 parents born, marking a groundbreaking leap in vitro fertilization, IVF. Now, this would be the first baby born of this technique in the United Kingdom, but it's not the first baby born in the world to this technique. The first baby, at least reported to be used, the technique that we're going to be looking at today, occurred in 2016 to a Jordanian family that was treated by a U.S.-based team that was working in Mexico, again indicating the limitations in approval of the process we're going to look at, as well as legalities. It's occurred in other regions of the world as well. There's a report of a family in Ukraine that used this technique as a way to treat infertility. If we're going to understand the science behind how we can use in vitro fertilization, basically an advanced in vitro fertilization technique, in order to create a baby with 3 parents, it's important to understand how in vitro fertilization works. So in vitro fertilization, in vitro is from the Latin, basically means in glass. So we're looking at doing this in an artificial setting, not really in a test tube, as the test tube baby kind of term was used early on in this procedure. But we're basically looking at fertilization, the bringing together of the male sperm with the female egg in a process that's occurring within the laboratory. So in vitro fertilization, what's occurring is basically a hormonal treatment is given to the mother or ultimately to the egg donor if that's the situation that's being used. The eggs would be ovulated, they would be collected, and then they would be combined in a clinical setting with the sperm to allow for fertilization to occur. Once that has happened, we're going to be able to have fertilized eggs that are going to go through a early stage of development, basically causing the cell to divide to produce lots and lots of more cells. And at a certain point, kind of this ball of cells that then could be transferred back into the uterus of the mother or the uterus of a surrogate mother for implantation of this embryo, this very early embryonic development structure, into the wall of the uterus, where it would then develop normally as if it had been occurring, the fertilization had occurred naturally within the body. Now, with the inherited disorder that we're looking at this week, it's a mitochondrial disease. And so in order to understand what a mitochondrial disease is, the first thing we need to know is, what is the mitochondria? And so the mitochondria is basically a small cellular organelle, which means that it's basically like the equivalent of an organ of your body, but it's an organ within a cell, an organelle within a cell, as opposed to an organ like your stomach, your heart within your body. And now the role of the mitochondria within the cell is to provide the energy of the cell. Now, many of us will talk about, eating, a good healthy meal to get the nutrients to power through the life. But in reality, our bodies, our cells don't use the glucose in our sugar or the amino acids in our proteins directly for an energy source. What's going to happen through a process of chemical reactions that occur to a limited extent within the majority of the cell, called the cytoplasm, the fluid of the cell, but very efficiently within these mitochondria, is a conversion of the food that we eat into a usable form of cellular energy. And so we need these mitochondria in order to convert the nutrients that we use, as well as burning the nutrients and the oxygen, to produce what's called ATP as a cellular form of energy. And so cells within our brain, cells within our muscles that use lots and lots of energy are going to be dependent upon having lots of these very efficient functional mitochondria to provide the energy that they need. Now, with this image from a 2018 article in Nature Reviews on Molecular Cell Biology, we can look at the different organs that are affected by mitochondrial disease. And again, the mitochondria are converting our food molecules, in essence, into a usable form of energy for the cells, so that cells that use lots of energy are going to have lots of mitochondria. And so we can see things like our skeletal muscles, which are going to be using the energy to shorten the muscle in the process of muscle contraction. Our brain, which uses lots of energy for the functioning of our nerve cells when we're thinking. Our heart muscles, which hopefully are going to be beating, contracting throughout our entire lifespan, are going to be using lots and lots of energy and are going to need lots and lots of mitochondria. Now, with these mitochondrial diseases, the amount of mitochondria that are affected and the extent of the damage to the function of the mitochondria varies, so that the disease itself can be varied from person to person. But in general, it is going to be associated with very significant impairments of function, and in many cases resulting in early death. Now, there's no cure for these mitochondrial diseases. In some cases, the symptoms can be treated using things like diet. And so nutritional management, maybe using a little bit of exercise and using supplements like vitamins and amino acids can kind of offset some of the symptoms, but they really aren't a cure for that disease. So if we look at how mitochondria are inherited, what we can see in this slide over here, again from the article Three's a Crowd from the U.S. Sun in May 2023, we can see these little blue structures here, and these blue structures kind of within this this drawing of the cell are going to be examples of mitochondria. And basically the mitochondria, they take up space. And so they're going to be found within the egg, which is a much larger structure, a much larger cell in this fertilization process. The sperm cell here is much smaller. It's still got the mitochondria to provide the energy for the tail to to propel the sperm forward to the egg. But the mitochondria coming from the father are going to be very limited in the effect that they would have on the infant. And so we need these healthy mitochondria in the egg in order for the infant, the child that's produced through the process of fertilization, to be able to have healthy mitochondria. Now, in the example we're looking at, the mother would have defective mitochondria, basically mitochondria that are not working properly, and these mitochondria would be passed on to the offspring, which would convey the mitochondrial disease to the offspring. And so with this procedure, what they're basically doing is taking the nucleus, this reddish structure right here. This has the DNA. This has the majority of the genetic information that's going to be coming from the mother. And that's going to be transferred into a donor egg. And so this would be another egg that has healthy mitochondria, healthy other organelles within the kind of mini organs, the mini structures within the cell. But what's going to happen is we're going to replace the donor nucleus, the donor egg nucleus, which has the DNA from the donor, and replace that with the mother's nucleus. So the mother's genetic information, the mother's DNA, the vast majority of the genetic information in the egg is going to be coming from the mother, and less than 1% is going to be coming from the donor. Through the process of fertilization, we are going to take that kind of modified egg here with the nucleus from the mother, the mitochondria from the donor, allow that to be fertilized in the in vitro fertilization process. We're going to get a fertilized egg, which will develop into an embryo, in which we're going to have DNA from the mother with the red nucleus here, DNA from the father delivered from the sperm nucleus, and then DNA from the donor, a very small amount of DNA from the donor in the form of this mitochondria. Now, this fertilized egg will develop for a limited amount of time within the clinical setting, and then it could either be stored by freezing it or implanted into the uterus of either the mother or the surrogate mother for development and hopefully birth of a healthy infant. Now, this process of basically nuclear transfer from the mother into the donor cell and using basically the donor's mitochondria and the DNA within the mitochondria is being looked at as an example of gene editing, because you're changing the genetics, you're changing the DNA of what would be this new infant that's being formed. And it's being described as a germline form of gene therapy. And so I want to review with you what's the difference between germline gene therapy, which has a lot of ethical considerations associated with it, as opposed to somatic gene therapy, which again has ethical considerations, but they're different ethical considerations. So in germline gene therapy, you're basically looking at altering the DNA or altering the genetics, the inherited material of the embryo very early on in development. And because of that, that results in all of the cells of that new individual, in this case the healthy baby hopefully, are going to have all of the DNA that have been altered in some way. Now, this includes what are referred to as germline cells, but basically the cells that are going to be either to produce the eggs in a woman or the sperm within a male. And so that genetic information that's been altered, it would be passed on to future generations. And so you have ethical considerations that if we were making a change to the genetics that would have an effect on all future generations, is that something that we should be doing? Now, the comparison to germline gene therapy, in which it's passed on to future generations, is somatic gene therapy. Somatic gene therapy, somatic is body, is basically looking at altering the cells within a region of the body. And so potentially treating cystic fibrosis, which is an inherited disease in which we can correct the DNA that codes for a protein within the respiratory system and counteract a lot of the effects of cystic fibrosis. But we're altering the DNA within a specific set of cells within a specific organ, and we're not affecting the production of the egg cells and sperm cells. So in somatic or body gene therapy, you're treating the cells of that individual, and that alteration of the DNA is not being passed on to future generations. But in this case, it's basically germline gene therapy because we're combining the DNA in the mitochondria from a donor with the DNA from the mother by transferring the nucleus, and then combining that with the father's DNA. And so we've got an altered kind of set of genetics that are going to be passed on to future generations in this process. So as I indicated on the previous slide, there are ethical considerations associated with this procedure. So to start out with, some religious concerns about the procedure. Looking at the Catholic Church, now they've never viewed in vitro fertilization as being acceptable because it removes the act of conception, the process of fertilization, from the marital act, because it's occurring not in the human body between a mother and a father as a loving act, but it's occurring in a clinical situation. And so in the 1987 doctrine of the faith called Donum Vitae, the gift of life, it basically says that medical technology or medical intervention that assists in the production of a healthy baby, healthy embryo, is acceptable if it adds to or assists with that marital act. But if it's done externally or it's done artificially, it's not accepted. Furthermore, in the process of doing this, the production of a healthy infant with the DNA from three parents, it results in the destruction of a fertilized egg involved in that nuclear transfer. And so there are ethical concerns associated with this. So looking at possible alternatives to this, very simply, one alternative would be the traditional process of in vitro fertilization using a donor egg. In this case, this would be a donor, and the pink individual here on the slide, providing the healthy eggs with healthy mitochondria that would then be combined with the sperm of the father to produce a healthy infant. But in this case, we'd have DNA and inherited traits from the father, but no maternal DNA, no mother's DNA, and no inherited traits from the mother's side of the family. So another alternative that may be possible under some circumstances is to go through the process of in vitro fertilization, but then prior to implantation, basically sampling the embryos in some way, and then screening them through pre-implantation genetic diagnosis to determine whether or not they have a high likelihood of producing a healthy infant. And so you would only implant in those embryos that had sufficient mitochondria to have function in that infant, and then discarding the abnormal ones. But again, this is a technique that may not be possible in this because of the variability in the process of the mitochondria and the extent of the damage to the mitochondria that may be present. And the final alternative, and again, probably not a good choice in this situation, is to allow for fertilization and conception to occur naturally, to allow the embryo to develop into a fetus, and so, many weeks of development, and then through the process of amniocentesis, basically inject a needle in, collect some fetal cells that are in the amniotic sac, the amniotic fluid, kind of surrounding the developing fetus within the mother's uterus, to take those cells out, to grow them in the laboratory in order to study them, and then do some DNA genetic testing to determine whether or not this baby that's developing within the mother's uterus has a genetic disorder, and in which that case there may be a decision made to abort or terminate the pregnancy at that point. So from that background, we come up with this week's ethical dilemma in science. Should in vitro fertilization use DNA from three parents to create a healthy baby? And so underneath that kind of main prompt, we can ask these questions. Is it appropriate to combine DNA from more than just the parents in order to create a baby without a genetic condition? Under what circumstances, if any, would it be appropriate to alter the DNA from a parent in order to create a baby without a genetic condition? And so basically looking at the first question is looking at using this nuclear transfer to alter the DNA in essence by combining mother's DNA with donor mitochondria. Is that acceptable? And then taking it a step further, saying, okay, under what circumstances could we actually go through and alter the DNA? So not just combining DNA from multiple sources, but to go in and possibly physically fix the piece of the DNA molecule that would correct the genetic condition that would be inherited. Thank you for listening to this week's Ethical Dilemma in Science. Please see the description below for additional information and references. So I've got a couple recommended articles that would get you kind of a good introduction into this process, as well as some additional references that if you're interested in learning about it further, you can dive deeper into the information. So thank you and have a great week.